Okay, today we're going to be examining antibodies and the generation of lymphocyte receptors. This is what Chapter 5 is all about. Now, first off, we're going to look at and be discussing the B cell as well as T cell receptors and how we can go from a small number of different receptors and having an expanded repertoire of different affinity receptors. The receptors may be for the same antigen, but they're going to have slightly different affinities, meaning they're going to interact stronger, they're going to interact weaker, and this will allow for selection later of the quote-unquote better or best B cells to make the best antibodies, or the T cells that will then have the greatest response to that viral infection and whatnot. What this is all going to be based around is gene rearrangement. We have a limited number of genes for this. Okay? But somehow these genes can be rearranged, and this is after the B cell has matured. This is going to be, the T cell during, growth in development in, the bone marrow. So we're going to go from a set of genes, few dozen, few hundred, depending on which ones we're talking about, and somehow we're going to be able to generate an antibody repertoire of 10 to the 11th. That's 100 billion billion different types of antibodies, each specific for a different antigen. Now, there's a couple of different ideas on how we were able to get to this, how we were able to generate so many different ways of coming up with these B cell receptors, which are exact copies of what the antibodies that B cell will be able to generate. The first one is what is referred to as the germline theory. The germline theory that each gene for each immunoglobulin chain is inherited. So somehow we've inherited 10 to the 11th different genes from this. Okay, there's just not enough room in our genome for that to be totally true. I'm not going to say it's completely discounted, because from what we've found as we begin doing genetic studies in the past few decades is there's also what is called somatic diversification. This theory says that this large repertoire of billions of different B cell receptors is due to rearranging certain gene loci, certain gene sequences. So yes, our germline theory does hold to an extent. We pass on in our DNA certain sets of genes that are responsible for the making of the B cell receptor, thus also responsible for the making of our antibodies. But we can't have 10 to the 11th different genes. What we have is a subset of genes for different portions of the receptor, thus different portions of the antibody, that will be rearranged as the B cell matures. As the B cell matures, they get rearranged. Here's a new B cell line. Later, when that B cell gets activated again and becomes a plasma cell, there will be a final level of gene rearrangement as we increase affinity. And this is also where the somatic diversification comes in. So germline theory to an extent, but most of what we're going to see for the extensive repertoire is going to be due to somatic gene rearrangement. We see that the immunoglobulin genes, the genes responsible for the making of the B-cell receptor, the genes responsible for the making of the actual immunoglobulin itself. Remember, the antibody is a protein. So it's going to start at the gene level, transcribe to RNA, translate to polypeptide. So if we go back to the base level, the genes, we can go through and do what is referred to as somatic recombination. We can take one from this subset, take one from column A, one from column B, and, two from column C, and there you go. You have a gene. Take, a different one from A, a different one from B, and just one from C this time. You've got a different. That's what somatic recombination is all about. Now, when we look at the genes and we trace them to the actual polypeptides, the actual protein, we see that there are different domains in the protein which correspond to different regions of the gene sequence responsible for polypeptide or the B-cell receptor production. The first one is what is referred to as the V domain. The V domain, it's going to have multiple segments, and somehow these segments are going to be responsible for the variable. Remember the different areas of the antibody? We had the variable, we had the constant domains. Well, the V domain is responsible for what we think of as the variable domains. Next, there's going to be a second region. This is going to be referred to as the J, or joining segment. And then followed by the D, or the diversity. So, when you look at what's going on here, DNA to RNA, RNA to protein. Okay, here is our central dogma. Germline DNA. This is at the stem cell level, okay, in the bone marrow. There's going to be some somatic recombination as the cell learns it's going to be a B cell, and it goes through some levels of recombination as it matures, and then as it becomes a plasma cell. So you're going to get VJ or VDJ, depending upon whether you're talking light or heavy chain, as we're going to see here in a minute. Rearrangement there, parts of the DNA cut out, others spliced together. Then you're going to get the primary transcript splicing that that you've learned in cell biology, genetics. Get rid of the get rid of the introns, keep the exons. mRNA gets translated, now you have a polypeptide chain. So when you look at the DNA, the double helix, what you see are different loci, L, V, J, C. There's some intervening sequences in between these. V, J, C. Cut, pasted together during somatic recombination. V and J go directly together. J links to C. And then you get our light chain. When you go through translation. The heavy chain, which has multiple constant regions, it's also going to have this D segment. So now it's VDJC. Again, V to D, D to J, or D to J, DJ to V, as you go through your levels of somatic recombination. The different C's are going to be joined together and then linked to DJ, LVDJ. Now, just because it's in a straight line here on the amino acid, remember this is nothing more than a one-dimensional strand. Once it goes through and starts doing its folding and this, that, and the other, we see the D and the J become part of the variable region up here where the B cell receptor is going to interact with the antigens. Just as we saw over here, J becomes part of the hypervariable regions. C's, well, this will give us our constant domains. Now, some of what we're going to be seeing here, where C is all of these different domains are brought together and then joined to DJ. Notice the AAA here. That means we're talking messenger RNA. So this is or the primary messenger RNA before RNA splicing. So you go through, cut out introns, exons, and then through RNA splicing, you join all the C domains together, and then you join all of C together to the DJ. Whereas up here, it was all at the DNA level. Now, what we have found is that many of these segments, the V, the D, the J, the C, actually have multiple versions. Light chain, heavy chain. Turns out light chains come in two forms, two flavors, kappa and lambda. Depending upon whether you're talking kappa light chains, lambda, remember it's the ratio of these. Mice had 20 to 1. We have, not quite that. Cows have 1 to 20, remember from chapter 5. Well, it turns out kappa light chains, the variable regions, has 34 to 38 versions. Each version is going to be slightly different. Lambda variable, 29 to 33. So they're pretty close in numbers, but there is some discrepancy there. The heavy chain, quite a bit more, 38 to 46. Since D is only in heavy chain, it's not in the light chain, there's no loci here for kappa or lambda, but there's 23 variants in the heavy chain genes. So that means that there's 38 to 46 V's to choose from. Choose one from that column. 23 to choose from in the D column. Anywhere from 4 to 6 in the J columns to choose one from. And then the constant. 1, 4 to 5, maybe 9 to choose from, depending upon whether you're talking kappa, lambda, or heavy chain. Now, not all of these are going to be functional. Okay, doesn't mean there are 46 H variable regions that you can choose from. Some of them due to mutations over time. You got to remember, we are collecting mutations. We are passing them down generation to generation. So some of these variants of the V are not going to be workable. They are not going to be able to translate into a functioning polypeptide that can have a secondary, tertiary, quaternary structure. We now refer to these as pseudogenes. They're still there. At the genetic level, they're still there, but they do not give a functioning V domain. These intervening sequences right in here, and these boxes, L1, L2, L whatever, you see here, the intervening sequences in between the J's, the D's, all of these are going to come into play, because embedded within them are specific DNA sequences, as you see with this heptamer, CACAGTG, or and then some space, and then what they refer to as a nonomer, ACAAAACC. These are what are referred to as recombination signal sequences, RSSs. These will allow for the different regions between the genes, between the domains, to interact and allow for the recombination to occur. But this can only work if the genes being recombined, or the domains of being recombined, are on the same chromosome. There is no crossover between chromosome 22 and chromosome 2 and whatnot. What we found is what they call the 12-23 rule. Turns out the turns, 1 or 2 turns, 12 base pairs, or 23 base pairs, will limit who can interact with whom. You look at the lambda chain. This V, lambda, is going to join with this J, lambda. The downstream sequence here is an RSS. Upstream sequence here is an RSS. 23, 12. Heptamer, space, nonomer. Nonomer, space, heptamer. These sequences are going to overlap. They are then going to be able to interact with each other and then allow for recombination. These RSS are acting as signals to other proteins that will then come in and aid with the recombination. Now, if you think back to Chapter 4, when I was lecturing on the hypervariable regions. The hypervariable regions at the tips of the antibody or the B cell Y. The hypervariable regions are CDR1, CDR2, CDR3. These hypervariable regions we find is that CDR1 and CDR2 is going to be embedded within the V sequence. For the light chain, CDR3 is going to be embedded in the region of the joining of V and J, whether it's lambda or kappa. For the heavy chain, CDR12 still in the V domain, but the hypervariable region, for it to form the CD3, you're going to have the juncture of V, D, and J together to get the proper folding to give you that hypervariable region. RAG1, RAG2, as well as recombinases will loop out our heptamer sequences, and then will allow that to be deleted, and this is now the germline. All cells that will come from this, all clones from this, will have only these genes, this V and that J. All the other V's are now cut out. There's also going to be a chance that instead of this direct loop, you're going to see this curly Q happening, which is going to insert both regions and will now allow for transcription to bypass all of these other promoters, and transcription will begin here at this V, at this promoter sequence, and continue on. So the V's are still there, all the V's, 1, 2, whatever, are still present, but they have now been bypassed. Here, they should point out whether you're going to do this direct loop or this curly Q looping is going to be determined upon the orientation of the V domain that you're going to be utilizing. Notice up here they're showing that they are in forward and reverse orientations. It is random which way they're going to go. So depending upon the orientation, this direct loop out or this curlicue. With this direct looping out, you're going to have a complete loss of all downstream V domains. Here it happens to be V1. So you lose 2 through 30 whatever. If this happened to be V10, then you would lose 11 through 30 whatever. But here, since it's in a reverse orientation, you're in your recombination, you're inserting this J directly here, so that now when this becomes resolved, you go V1, V2, you hit the heptamer nonamer, and you see there's been a recombination, the changing of the direction of that V domain that is directly attached to that J, and then it goes downstream to the C. How's this work? What is the actual mechanics going on here? Excuse me. Well, as I said, you have RAG dependent VDJ rearrangement. If you were talking heavy chain, you're going to have a version for the light chain that we just talked about. RAG 1, RAG 2 are going to bind to our nonomer and our enantiomer, our 2 signaling sequences. So when you look a little closer here, you see RAG1, RAG2 binds the 23 number here at the downstream of the V, which brings into close proximity and binds to the 12-mer upstream of the J, holds them in close proximity so that they can be cut. And then you have an open ends. Now, what I don't expect you to know is Q70, Q80, which are part of this big VDJ recombinase, holds the V, holds the J sequence. Then you have its, complement, Q70, Q80, holding the 23-mer, 12-mer. So that here, the coding joints held by Q70, Q80, the other parts of our recombinase, the DNA, PK, Artemis, are going to begin processing, and then you're going to see TDT and other enzymes are going to fill in the double-strand breaks. Fill in the double-strand breaks, precise coding, so that you have VDJ. The remaining over here, you're going to have, again, a ligase coming through, do a fixed double-strand breakage, the releasing of Q80, Q70 for, so they can be recycled, and then all this intervening stuff that was there is now lost.